Hello. Good morning and good afternoon to everyone, and a very warm welcome to all the participants of this first webinar of the Webinar 2024 series. I am Shivendra Kishore, the CEO of our Census, and also coincidentally your host for today's session. I hope you are also equally excited as myself to listen to the great speakers of today. The topic of this webinar revolves around mitochondrial diseases and the testing strategies. The topic has been carefully selected based on numerous queries that we receive at our Census. So please help me in welcoming the speakers of today, Dr. Alison Faber and Dr. Muath Al-Qurashi, who bring a wealth of experience in this field and also a unique perspective into how to deconstruct the genetics behind mitochondrial disorders. The speaker of the first half will be Dr. Faber, who is a seasoned geneticist, groomed in the clinical environments of Centogene, Blueprint Genetics, and now as well our Census. She's currently leading the customer relationship and product management team at our Census and is constantly interacting and collecting feedbacks from doctors and patients. Dr. Faber will present the genetics and diagnostic testing strategies of the mitochondrial disorders. And I'm also very delighted and honored to introduce our guest speaker of today, Dr. Muath Al-Qurashi, a distinguished pediatric neurologist based in Jordan. He's also a strong proponent of whole genome sequencing as a first line of testing for genetic disorders. With a career spanning over 20 years, he continues to see numerous children with neurological disorders in his clinical practice. And Dr. Muath will provide us insights on how to identify patients with mitochondrial disorders and will present us clinical pictures and some case studies of patients diagnosed here at our Census. Just as a note, participants who would like to interact with the speakers through questions or comments, please type your questions in the chat box on the bottom right, and we'll pass it on to the speakers during the Q&A session. There will also be a polling session where we will randomly test the familiarity of the audience with the topic and collect some feedback, so please stay tuned. The webinar is being recorded, and as a tradition of sharing knowledge, for participants who have registered and may have missed a part of the webinar, we'll be sharing the slides as a handout after the end of the session. So now, without any further delay, I would like to hand over the stage to Dr. Alison Faber.
All right, thank you. And thank you everyone also for joining. It's lovely to be here and talk about mitochondrial disease. So I want to give a little overview of what I hope to accomplish in my section and in the talk generally. I'm going to start by describing the features of mitochondrial disease and then define the value of the molecular diagnosis and kind of outline how genetic variants can cause mitochondrial disease. Then after that, I'll hand it over to Dr. Muath, and he will describe 3 patient case studies for you. All right, I want to start really simply with the definition of mitochondrial disease. This is any member of a large group of disorders that are caused simply by mitochondrial dysfunction. So this can be a dysfunction in the energy production pathway, so what we'll call a primary mitochondrial disorder, or any dysfunction in the integral mitochondrial function, so anything that sustains the mitochondria, which we'll refer to as a secondary mitochondrial disorder. So I want to stress straight away that mitochondrial diseases are among the most genetically and phenotypically diverse group of inherited diseases, which makes them complicated to identify in the clinic and diagnose. So I want to start here straight away with our first poll question. So we'd like to right away kind of get a feeling for who is in the audience. So the question on your right, if you click on polls, will be, have you ever suspected a mitochondrial disease in your clinical practice? And I will give you a minute to choose one. Okay, so we'll give it one more second, but we see the majority of answers have come from people who aren't physicians. So maybe geneticists or people just generally interested in mitochondrial disease. So that's awesome. We can all learn together some basics. Oh yeah, and a few physicians who have already seen mitochondrial patients. Okay, thank you for participating in our poll. We'll move on. I want to come back to this idea of phenotypic diversity and what exactly I mean by that. So this slide looks busy, but because mitochondria can be found in every cell of the body, dysfunction in the mitochondria can affect each organ or tissue type in a variety of different ways. So this figure on the right is showing how mitochondrial dysfunction can impact different tissues in the body. So for example, in the eye, this can look like optic neuropathy or retinopathy. In the heart, this can be conduction disorders or cardiomyopathy. Commonly in skeletal muscle, we'll see weakness, fatigue, and myopathy. In the brain, this can present as seizures, stroke, or dementia, but mitochondrial dysfunction can impact all of the organs: the colon, the inner ear, the blood by the way of bone marrow, pancreas, kidney, and liver. And all of these symptoms can vary from mild to severe. They can be present at birth or have an onset at any age, which makes the identification of these things pretty complicated. This figure on the bottom, I've shown some common mitochondrial diseases and their approximate ages of onset. So we can kind of see this wide variety from Barth syndrome that would be present in a newborn to syndromes like Lee syndrome or Alpers syndrome that could have onsets in childhood, or all the way up to progressive external ophthalmoplegia and optic neuropathy, which usually have their onsets in adulthood. So each of these syndromes that I've shown here are rare diseases. But if we think about mitochondrial diseases generally, they're relatively common. So more than 1 in 5,000 people in the world has a genetic mitochondrial disease. And as we showed in the last slide, this can affect any organ. So this means that a physician in any specialty has a pretty good chance of coming across a patient with an underlying mitochondrial disease. Now, while there's no cure for mitochondrial diseases, there are treatments that can ease symptoms and prevent life-threatening complications. So it's extremely important that we identify the underlying molecular etiology or genetic cause of primary mitochondrial disease to really tailor these treatments to each patient. So how can we do that? First, we need to be able to identify a potential mitochondrial disorder in the clinic. So I've listed 3 features that would indicate an underlying mitochondrial disease. The first are the clinical features. So mitochondrial disorders should be a differential diagnosis for any progressive multisystem disorder. In children, this can look like poor growth, muscle weakness, developmental delay, and sensorineural hearing loss. In adults, this looks a little different. Usually it could be ataxia, progressive external ophthalmoplegia, dementia, myopathy, optic atrophy. So you see that this is a wide variety of symptoms. And also importantly, none of these symptoms is completely unique to mitochondrial disease. So even when we suspect a mitochondrial disorder, there could be other diagnoses that are being considered. The second feature is family history. So any pedigree with multiple affected members with similar symptoms in multiple generations is a good indication of an underlying genetic disorder generally. Specifically, a clear maternal inheritance pattern can indicate a mitochondrial disorder, but it isn't a requirement for mitochondrial disease. So finally, there are very good clinical test results that can indicate mitochondrial disorders. This can be muscle biopsies or biomarkers like lactate and pyruvate in the blood, or the cerebrospinal fluid or urine, or oxidative stress markers are very good indications of mitochondrial disease. So to understand how dysfunction in the mitochondria can cause this wide variety of phenotypes, I want to take a little detour into cell biology and talk about mitochondria for a few minutes. So these are organelles that generate energy for the cell. So if you look at the figure on the right, the mitochondria are these orange bean-shaped organelles. They have an outer membrane and an inner membrane. And it's this inner membrane where we have this 5-step or 5-complex system for generating energy. Now, just like in the picture, each cell can have multiple mitochondria depending on its energy needs. And most importantly, for what we're going to talk about, is these mitochondria, like all organelles, are inherited maternally, so from the mother, which is important to the mitochondria and unique to them because they're the only organelle that contain their own genome. Each mitochondria has around 10 copies of this circular genome that contains 37 genes, always inherited from the mother. So pretty unique. So how do these genetic variants end up causing mitochondrial disease? I want to start by stressing that primary mitochondrial disorders are genetic disorders. When we have a diagnosis of primary mitochondrial disorder, there is always a known or presumed underlying genetic variant responsible. There are more than 1,500 genes that encode mitochondrial proteins. But I just told you in the last slide, there's only 37 genes in the mitochondrial genome. So this figure on the right illustrates nicely how the genes in the nuclear DNA contribute proteins that are necessary for the mitochondrial function. So up in the left, we have a nucleus with some nuclear DNA, and those genes are being translated into proteins in green here that are important for the actual energy production pathways, as well as maintaining the mitochondria itself. And then if we follow this brown pathway, the nuclear genes are making proteins that are required for the translation of mitochondrial DNA genes and the replication of the mitochondrial DNA. So if we consider the nuclear DNA and the mitochondrial DNA, variants in more than 350 different genes are known to cause mitochondrial disease. And these numbers are growing constantly as new research is being done and publications are released. So disease-causing variants can be inherited from either the nuclear DNA or the mitochondrial DNA. They can arise de novo or spontaneously in an individual in either, or in the mitochondrial DNA, they can be acquired. At this point, I would like to go back to the polls, and we have a quiz question, number 2, to find out if I have explained this concept accurately. So quiz question 2 is mitochondrial diseases can be caused by variants in the mitochondrial DNA, the nuclear DNA, or both. So I'll give you 1 minute. Weigh in. Okay, so we've got some answers rolling in. All right, so the correct answer is both. And we're about half and half of the mitochondrial genome. So this is a common misconception, and I'm glad I'm glad to see this poll. So if we look back at the figure, the genes in the nuclear DNA are essential to the function of the mitochondria and the production of energy. So variants in nuclear DNA can cause dysfunction in mitochondrial proteins and cause mitochondrial disease, as well as those 37 genes in the mitochondrial DNA. So that's a really essential point. Okay, let's move on. Thank you guys for participating in the poll. So I want to talk a little bit about variants in both nuclear DNA and mitochondrial DNA. I want to start with something a little bit more familiar, which would be the nuclear DNA. And most primary mitochondrial diseases are caused by variants in the nuclear DNA. And most of those are inherited in an autosomal recessive manner, which means that we need a copy of the variant in both genes to cause disease. But mitochondrial disease can also be inherited in an autosomal dominant manner, so where one copy of the gene has a variant, and that's sufficient to cause disease. Also, genes on the X chromosome can cause mitochondrial disease in a recessive and dominant manner. And so what I've shown in this figure is that when we talk about these nuclear DNA gene or variants, we could discuss them as homozygous, where the alleles are the same in both copies, heterozygous, where the alleles are different, so one copy and one wild type, or hemizygous, so a male on the X chromosome, where they have one copy of the variant, which is all the copies that they have. So when we think about nuclear DNA like this, the reason I'm bringing it up is that when we discuss mitochondrial DNA, we don't have the same terms because each cell does not contain 2 copies of all of the mitochondrial DNA. So as I mentioned previously, the mitochondrial DNA is a ring. Each mitochondria contain multiple copies of the mitochondrial DNA, and each cell contains multiple mitochondria. So therefore, we have a lot of copies of this mitochondrial DNA in each cell. So instead of thinking about we think about this as homoplasmy and heteroplasmy. So homoplasmy would be where all of the copies of the mitochondrial DNA are exactly the same. And heteroplasmy is the presence of two or more types of mitochondrial DNA in the same person. So we're thinking about this as a percentage of mitochondrial DNA that contains the variant of interest versus all of the other mitochondrial DNA. So a little bit different than how we think about the nuclear DNA. Okay, now I want to talk about how this heteroplasmy arises and how it can be so different. So there's two ways that cells can end up with mitochondrial heteroplasmy. Inheritance from the mother. So in this figure on the right, we're showing the oocyte maturation from primordial germ cell to mature oocyte. And variants in the mitochondria are in red and wild type mitochondria are in green. So if we start here, there's a selection process in oocyte maturation that can result in mature oocytes with different levels of heteroplasmy than the original primordial germ cell. So these then mature eggs can be fertilized and become embryos. And at that point, the second cause of heteroplasmy can take place, and that's somatic mutagenesis. So during embryogenesis, a de novo variant in the mitochondria can arise. And at this point, we have an embryo with some level of heteroplasmy. During the cell division of embryogenesis, we can have a skew, an unequal distribution of mitochondria containing the variants. So we end up with different levels of heteroplasmy in different tissues as that embryo develops. So that is my first implication of this heteroplasmy levels, is that different tissues in an individual's body can have different levels of heteroplasmy. Now, this is an important thing to keep in mind, not only for sampling, but for disease threshold. In order for a cell to be symptomatic, there must be a certain level of mitochondrial variance and therefore mitochondrial dysfunction that's reached. That's the disease threshold. Now, this value can be between 30 and 90% heteroplasmy. And this will depend on the tissue type we're talking about, the gene in which the variant is, so the function of that gene, and the specific variant itself will determine the exact level of that threshold. Now, the last implication that I want to point out on this slide is that if we imagine that each of these mature oocytes is fertilized and now becomes three individuals, three siblings, we can imagine that the siblings may have inherited a variant and still have different levels of heteroplasmy from each other and from their mother. Now, this can make creating that pedigree and finding that maternal inheritance very difficult and can make segregation analysis very difficult. So just an additional complication to keep in mind. And because this is so complicated, I have created our final poll question. So question number 3. Yep, now you should be able to see it at the top. So, will a patient's heteroplasmy level always match their mother's if they've inherited the variant? I will give you a minute to weigh in. Okay, thank you for voting. We've already got a great number. Yes, and the answer is false. Even when a variant is inherited, an individual can have a different level of heteroplasmy identified than would be in their mother. And also a variant could be always de novo as well. So thank you for participating. I'm delighted. All right. So now that we've established that mitochondrial diseases are difficult to identify in the clinic and the underlying genetic causes are a little more complicated than variants that can exclusively be found in the nuclear DNA, I wanted to discuss the types of genetic tests that you could order when you suspect a mitochondrial disease. So I've listed 4 sequencing tests here, from kind of smallest to largest. So the first would be a test where we only sequence the mitochondrial genome, just the mitochondrial DNA. The second test would be a mitochondrial disease panel. This would include that mitochondrial DNA and all of the genes in the nuclear genome that are known to cause mitochondrial disease. Larger than that would be a whole exome sequencing. So this is a situation where we would sequence the mitochondrial DNA and all of the protein coding regions of all of the genes in the nuclear genome. And finally, the largest test would be whole genome sequencing. In this situation, we would sequence the entire mitochondrial DNA and the entire nuclear genome. So what are some different situations in which you would order each of these tests? So I have use cases here for 3. So ordering just sequencing of the mitochondrial DNA could be a good option for a patient with a very clear clinical symptoms and a family history with clear maternal inheritance. So I've already mentioned that most mitochondrial diseases are caused by variants in the nuclear DNA, but if you have a patient with where we can see that maternal inheritance, and that's a really good clue that the variant we're looking for is in the mitochondrial DNA, and we're not considering any non-mitochondrial diagnoses. The example would be Leber hereditary optic neuropathy. 95% of those cases are explained by variants in one of 3 genes in the mitochondrial genome, and it's a very specific phenotype. So then maybe a mitochondrial DNA sequence would be a good test. So in the absence of that maternal inheritance pattern, when you have a patient with a very clear symptoms indicating mitochondrial disease, this would probably include some clinical lab results. So you're not considering other diagnoses, then a mitochondrial disease panel might be a good option. Now, the final category is a patient with complicated clinical features or manifestations that are unclear or atypical. So this is where you would probably suspect a mitochondrial disease, but you wouldn't be able to rule out other syndromes. This is when a whole exome or whole genome sequencing would be a great option. So no matter which genetic test you choose, when you suspect mitochondrial disease, the recommended sample type is going to be blood or buccal swab, so something relatively non-invasive for a first-line test. Because I've listed whole exome and whole genome sequencing together in this use case slide, I wanted to take a minute to explain what the differences were between those 2 tests. And the essential difference is the types of variants that we're going to detect. So in this table, I've compared the whole exome and the whole genome by the types of variants. So as I mentioned, whole exome includes the sequencing of the protein coding regions of all known genes. So in both tests, we're going to be able to detect the single nucleotide variants and small insertions and deletions in all of those exons. We're also going to sequence the mitochondrial DNA in both tests and detect variants equally well. We're going to find variants that are as low as 5% heteroplasmy. Now, where these 2 tests start to differ are intronic variants. In the exome, we're only going to detect intronic variants that are very near the exons, whereas in the genome, we're going to sequence the entire introns and find all of those variants. And finally, and most importantly, I would say whole genome sequencing is very good at finding all of the structural variants in the nuclear DNA. So these are large insertions and deletions and rearrangements that whole exome sequencing is not going to be able to identify. So no matter which of these sequencing tests you choose, and if they're mitochondrial DNA or nuclear DNA, all of the variants are going to be classified based on their likelihood to cause disease using the ACMG guidelines. So this is where we take all of the evidence that is known for a variant and determine if we are sure that this variant will cause disease and classify it as pathogenic, or we're sure that this variant cannot cause disease and classify it as benign. If we're kind of certain, we have plenty of evidence, it could be likely benign or likely pathogenic. And all of the other variants that don't have enough evidence for us to predict if they're causing disease or not, we're going to classify in the middle as variants of uncertain significance. So specifically to mitochondrial disorders, those lab tests that I mentioned previously can be used as evidence to classify variants. So they're very important to do those tests and provide that information when ordering a genetic test. All right, so I have told you some tips for identifying mitochondrial disorders in the clinic and some genetic tests that are available. So I will ask the last poll question for my section. Number 4, at this point, would you be comfortable recommending a genetic test if you saw a patient and suspected mitochondrial disease? So we have yes, no, I don't know, maybe for those of us that aren't seeing patients, or if you have already ordered a genetic test, you can choose the last option. Okay. Well, that's fantastic. we're well into the yes or have already section. So that's great. I am going to hand it over at this point to Dr. Moath to show us our 3 case studies, and then afterwards, both of us can take your questions. Okay, thank you.
Hello, everybody. So let's start with our first case. Before starting with the first case, I would like to emphasize some points which are important for us to understand. So mitochondrial diseases is highly heterogeneous. As we used to hear, it might present with any symptom in any organ at any age by any mode of inheritance. Second point important to remember is there is no common biomarker to easily detect mitochondrial disease or dysfunction in all cases. And third one, and also important, there is no single common genetic cause for mitochondrial diseases. Rather, it's now recognized that mitochondrial diseases is the result of inherited mutation in any of the, as Dr. Allison said, 37 genes of mitochondrial genome, or the almost now more than 250 genes in the nuclear DNA. So, by this introduction, I want to emphasize that it is many of the time it's difficult to diagnose mitochondrial disease in any patient without going to further investigation and especially the genetic testing. In a cohort of 250 patients with confirmed mitochondrial disease, the top five common symptoms were muscle weakness, chronic fatigue, exercise intolerance, GI disturbance, and balance problems. None of these symptoms is specific for anything. So it might present with any other disease. And keeping in mind, when dealing, at least in my daily practice, dealing with pediatric patients, many times the patient present early in the course of the disease, and there are no clear symptoms toward specific diagnosis. So putting everything together, it emphasizes the important role of genetic testing in these unclear, non-specific cases in order to reach the diagnosis earlier in the course of the disease. And as we all know, without a diagnosis, no treatment plan, no obvious treatment plan. Treating the symptom is completely different than treating the disease itself. Knowing its complications, knowing its implication on the family planning, and cutting the cost, which is also very, very important. With the advance of the next generation sequencing, they gave us day by day important tools. It helps us in our daily practice. It's also make the diagnosis easier for us, and it costs less than what was expected in the beginning. Let's talk about our first case. This is a 12-year-old girl. She's the only child of relative parents. The reported symptoms were thyroid nodule, abnormal eye movement, horizontal gaze palsy, abnormal saccadic eye movement, and ophthalmoplegia. Non-specific symptoms. The previous whole exome sequencing performed at another lab, and it was negative. Next, please. So, this is the result of the whole genome sequencing. came back positive. Heteroplasmic, almost 55.4% deletion of the mitochondrial DNA. This deletion includes 17 genes involving in complex 1, complex 4, and 5. The deletion could be detected with any test that include mitochondrial DNA sequencing. So ordering a whole exome sequencing alone, it was a waste of time, at least for me. Next, please. Main form of mitochondrial disease caused by, as we saw, a single large-scale mitochondrial DNA deletions. These are the three main forms: the Kearns-Sayre syndrome, which manifests with progressive external ophthalmoplegia, onset before 20 years of age, pigmentary retinopathy, cerebellar ataxia, heart block, deafness, myopathy, dysphagia, diabetes mellitus, and hypoparathyroidism, dementia. Pearson syndrome with sideroblastic anemia of childhood, pancytopenia, exocrine, pancreatic failure, renal tubular defects, and the progressive external ophthalmoplegia with external ophthalmoplegia, bilateral ptosis, and mild proximal myopathy. So, single large-scale mitochondrial DNA deletion, never inherited. These disorders are typically caused by a de novo deletion that occurs in the mother oocyte during germline development or in the embryo during embryogenesis. Next, please. The message from the first case. If the proper tests were ordered from the beginning, it will cut the cost, the effort, and it will be more comprehensive to cover these symptoms from the beginning. So doing a WIS alone is not enough at all for diagnosing the first case. As Dr. Allison said, it covers just only the exonic part of the gene, doesn't cover the mitochondrial genome, and sometimes it takes 4 to 6 weeks in order to come back the results. Then doing another test, again, more cost, more wasting time. The second case is a 2-year-old girl with non-consanguineous parents, one healthy sister, maternal cousin with coarctation of the aorta since childhood, paternal grandmother with stroke and hypertension. So again, the family history were non-specific. The reported symptoms: abnormal corpus callosum morphology, developmental regression, failure to thrive, global developmental delay, intrauterine growth retardation, microcephaly, anemia, elevated alpha-fetoprotein, elevated hepatic transaminase, recurrent hypoglycemia, increased serum lactate, metabolic acidosis, bruxism, dehydration, recurrent infection, and central hypotonia. Again, multi-system involvement. In this case, you might go straight forward and think that multi-system involvement, a little bit of lactate elevated, we might think about mitochondrial disease. So what tests to be ordered? Next slide, please. So again, whole exome sequencing performed at another lab, and it was negative. It showed 3 variants. All of these variants were classified as a variant of uncertain significance, not explaining the case. So next step, what to do? Again, go for whole genome. Whole genome sequencing. Here we have an interesting result. The result showed same VUS in the same variant, which were classified as variant of uncertain significance, and this variant was reported in one patient with Lee syndrome in trans with another pathogenic variant. The whole genome, which include the intronic part of the gene, showed that we have another most likely pathogenic variant. It's deep intronic variant, and functional studies have shown that the variant impacts splicing. So, detected and reported to be detected in a homozygous state in 11 individuals with Fanconi syndrome. So, this variant could only be detected by whole genome sequencing. Next, please. So, this variant in this gene are associated with mitochondrial complex 1 deficiency, nucleotide 17. Okay. These are the common symptoms, which is hypotonia, flat feet, scoliosis, gait difficulties, developmental delay, dystonia, dysarthria, rigidity, ataxia, lactic acidosis, and increased serum lactate. By ACMG guideline, we are not supposed to use VUS in clinical decision making, but clinical decisions should be based on personal and family history. So in this case, in order to confirm the diagnosis, we need to go for the sequencing for the same variants in each parent. If one variant is inherited from each parent in trans, not in cis, the variant can be reclassified from VUS to likely pathogenic. So this is another usual case in which whole exome sequencing did not reveal any disease, but by whole genome, by looking into the depth into the gene, the intronic part, it gave us at least a possible diagnosis which need to be confirmed by examining both parents. Next, please. So this is our third case. This is another interesting case. It's my patient, actually. She is a 9-year-old girl, non-consanguineous parents. They have a lot of cancer running in the family, and twin died at birth. Now, the reported symptoms were aggressive behavior, global developmental delay, hirsutism, hyperactivity, and short attention span. The interesting part in this young lady was she was diagnosed as ADHD, non-specific developmental delay. For her hyperactivity, they started her on multiple medications. One of the doctors who saw her, he thought that she had some sort of epilepsy, seizure, whatever, and started her on sodium valproate. As we all know, in mitochondrial diseases, there are some drugs you need to avoid. One of these drugs is valproic acid, which is a common and easy to describe everywhere in the world anti-epileptic drug, anti-seizure medicine, whatever. The family were putting her in excessive physical treatment, pushing her toward academic performance, trying multiple drugs in order to improve her performance without knowing what is the diagnosis. The most important thing that you can see here is not the behavioral issues, it is the developmental delay, the unexplained developmental delay. She has poor fine motor skills, her gait is unbalanced, so there were many red flags in this case. I stopped everything from her medicine and I ordered a whole genome sequencing. Next, please. So, came back positive for heteroplasmic variant in the mitochondrial TL1M at the position of 312, etc. This pathogenic variant is responsible for 80% of MELAS cases. MELAS, as you know, a genetically heterogeneous mitochondrial disorder with a variable clinical phenotype. And the common symptoms are myopathy, encephalopathy, lactic acidosis, stroke-like episodes, changes in behavior, hearing loss, and developmental delay. Actually, she had some of these symptoms present in her, and now we have a diagnosis. And this diagnosis will impact our treatment plan and will change our future family planning for the family. And this variant could be detected with any test that includes mitochondrial DNA sequencing. However, the symptoms were not indicative of mitochondrial disease. So sometimes you have this patient with a vague clinical picture, and you are not expecting the mitochondrial disease, and voila, it is there. So keep in mind, mitochondrial disease is very, 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 very tough to diagnose. And the only way is to go and to have a high index of suspicion, to examine your patient from A to Z clinically, do some biomarkers, it might help you, consult other colleagues, ophthalmologists, GI, whatever, in order to gather more info and then order the proper test. If you are suspecting something and you have no solid evidence to do specific test, please go from the beginning, save time and money of the patient, and order a test that is comprehensive. It covers all aspects of the genetic diseases that we know, and it will cut many of the effort of the patient and the doctor and the family. And next, please. So mitochondrial diseases are the most genetically and phenotypically diverse group of inherited diseases and can arise at any age. Red flags include progressive multisystem disorder, family history, and clinical test results. Most primary mitochondrial diseases are caused by variant in the nuclear DNA, but can be caused by variant in the mitochondrial DNA as well. Heteroplasmy is the presence of 2 or more types of mitochondrial DNA in the same person and can have implication for the diagnosis. Genetic testing is essential for diagnosing and treating mitochondrial disease. Thank you. Any questions? We are ready to answer. Yes. Dr. Alison.
Yes, so thank you. Yep, we'll take questions, and there's 2 kind of like summary poll questions at the end. If you could answer those 2, we would like to see that. So I saw a question that asked, why in case 1 did the whole exome sequencing not detect that heteroplasmic deletion? And we did not get the results of that test from another laboratory, only the statement that it was negative. So I assume that the other whole exome did not include the mitochondrial DNA.
Definitely. Yeah. It's a simple clinical WES.
Exactly, yes. So if you're ordering a WES and you're suspecting mitochondrial, make sure it includes the mitochondrial DNA. Yes.
Alison, what would be the average coverage recommendations for reporting mitochondrial variants?
Oh, when the mitochondrial DNA is sequenced, there's on average at least 1,000 reads, so 1,000x coverage of that of each position, and we can reliably report variants that have heteroplasmy of up to 5% in any tissue type. So yeah, so the variant is being detected at a very high rate. Am I understanding the question correctly?
Mm-hmm. There are a lot of questions around, so I'll pick one. Which is the recommended reference genome for mitochondrial analysis? Like we have HG18, HG19. Is there a reference? Because I think mitochondrial reference genome must have been, to my understanding, very standard and not much changes are happening. But is there any recommended reference genome is the question.
I don't know the answer to that question, but I can find out. Yeah,
because
that's a good question.
To my understanding, every HG19 or HG38 includes a mitochondrial. So I would say we should take the latest reference assembly.
Yeah, we do use the latest reference assembly, but I think that the mitochondrial genome reference doesn't change as much as the nuclear DNA is improved with each release. So
there is one question which says, how nuclear genome influence mitochondrial genome? So perhaps a little bit rephrasing it, I would ask, can one distinguish based on symptoms and age of onset if the mitochondrial disease is originating due to mutations in nuclear DNA or in mitochondrial DNA? So this may be perhaps to Dr. Muath.
Yeah. Usually, nuclear DNA-related diseases present earlier in life. In recent studies, it shows that, let's say, about 80 to 90% of nuclear DNA-related disorders present in early infancy and later on. Then, as the age goes later it become more of with aging, become more of mitochondrial DNA. But recent studies had showed that with widespread of sequencing, that both are present in almost 1/3 to 2/3. So both might be present at birth and later on. So depending on the age alone is not actually not a good way to differentiate between is it nuclear or it's mitochondrial. So again, back to point number one. If you suspect, order something comprehensive to cover all possibilities. There is no common biomarker. There is no single common genetic cause.
Okay, thank you. There's one question related to case 1 with single large-scale deletion. Which tissue DNA was sequenced? Large-scale deletions are mostly not detected in blood DNA, therefore muscle DNA is preferred. Can tests for large-scale deletions, single and multiple, be ordered at our sensors? And adding to that is also another question where somebody asks, since mitochondrial mutations affect multiple organ systems, our symptoms are very heterogeneous, how do we identify the exact tissue type in order to avoid repeating an NGS test from blood and then again in a specific tissue type? Alison, would you take that?
Yes, the first one, the first part.
I'll take the first part. Yep. So case 1 was blood. And in people under the age of 18, blood is a fantastic tissue type to test for even those large scale deletions. There should be no problem in detecting mitochondrial variants in blood. It is recommended generally that if you have any patient under the age of 18 and a mitochondrial suspicion, start with blood or buccal swab. A less invasive test will be a very good stage, even though there might be higher heteroplasmy in the muscle and affected tissue. That's why we report as low as 5% in the blood, even though that might not reach the disease threshold. It'll be higher in the affected tissue, that heteroplasmy level. So, yeah, perfect. Do you want to say that as well?
Dr. Moat, your comments, please.
Yeah. Regarding what tissue, it depends on the clinical picture, on the common biomarkers that we have done. The progression of the symptoms, everything will gives you an indication where to go. Now we usually go for blood or saliva, whatever. Blood is the preferred sample. If it is confirmed by sequencing the blood sample and everything is sound and clear, it's okay. In some cases where we are suspecting a mitochondrial disease and we have symptoms that, especially in adults with myopathies or so on. Sometimes you do the genetic testing and the result is inconclusive or negative, and you're still suspecting that there is a mitochondrial disease. At that time, you might go further and do the muscle biopsy, you might do the skin biopsy, you might do something more specific and do further investigation by skin or histopathology, and sometimes you might go for mitochondrial sequencing through this affected tissue. These usually nowadays, it's really, it's less than 5% of these cases. We go for specific tissue in order to do the sequencing of that. But usually most of our cases can be solved by blood sampling and sequencing the blood sample.
Thank you. Is there any algorithm for testing the mitochondrial disease? So something that people could rely on?
Yeah. Again, we will start from the beginning. Clinical examination, pedigree analysis. We will do some phenotyping of the symptoms. After that, we might go for some simple blood tests, biomarkers like lactate, CK, ALT, looking for any markers that might indicate something. After that, we need to confirm. Actually, these days, you need to do genetic testing. You need to do a test that makes, it covers everything from the beginning. After this point, if the genetic testing is okay, it's positive, you go for standard clinical care let's say trials for treating this disease. Now, if the genetic testing is inconclusive, then you go for further testing, as we said, for specific tissue biopsy and so on. Still, most of our data will be taken from the clinical examination, from the family pedigree, and from the phenotype of the symptoms. What do you mean by that? If a child with, let's say, white matter disease, developmental delay, you do an MRI, and this MRI is clear cut, no way to go back. This is a hypoxic ischemic injury. No need to go for further. But if a child with developmental delay, you do an MRI, and this MRI has non-specific finding. He has some visual issues, hearing loss. This case is progressive. Don't waste money and time repeating another MRI, repeating another blood test, and so on. Go for genetic testing from the beginning. The clinical picture is vague, multi-system involvement, so go to the target, go for genetic testing. But in some cases, when the clinical history, matches the clinical findings, like in cases of, let's say, autism or whatever, in hypoxic injury, in hemorrhages, whatever, no need to go for further. So you need to match everything by clinical judgment. If you are convinced as a physician that this case goes with that diagnosis, it's okay. If you have doubts or you are not sure, please go for genetic testing.
Thank you, Dr. Moat. One question which is following the question that we asked now, is there any treatment available, or in future would it be possible to treat mitochondrial disorders through perhaps gene editing, CRISPR? And what are the patient management plans would you recommend when you identify patients with mitochondrial disorders?
So we all know about the mitochondrial cocktail. These are multivitamins, supplementations in order to improve and to maintain the function of the mitochondria. We usually give our patients, once they are diagnosed with mitochondrial disease, we give them the coenzyme Q10, the riboflavin, the arginine, the alpha-lipoic acid, the folinic acid, and the carnitine. All of these are supplements to support the function of the mitochondria. This is the usual and the classic mitochondrial cocktail that we are all using in our daily practice. Now, the important thing in managing this patient is, first of all, as we said in third case, to avoid drugs that might exacerbate the symptoms or might causes an overstress for a patient with low level of energy in order not to deteriorate. So this is important regarding choosing other drugs for other, as we all know, mitochondrial diseases are multi-system involvement, many symptoms, you need to use many drugs. So it helps you in choosing and putting a treatment plan that will not affect the patient. What else we do for mitochondrial patient is mitochondrial disease is we might go for exercise to improve muscle fatigue, the function of the quality of life, and so on. And even though so, we advise for exercise. We need to stop exercising during viral illnesses or during fasting. And our goal is to preserve the strength and the mobility of and the functioning of muscles and in general well-being. There are some talks about ketogenic diets and the mitochondrial diseases, especially in cases when we have refractory epilepsy and mitochondrial disease. There are some reports, they are not that solid, but it's advice for using the ketogenic diet for patients with refractory epilepsy and mitochondrial disease. Apart from it's contraindicated in mitochondrial DNA deletion myopathies. It's preferred not to go for the ketogenic diet. Other things is during if we go for any kind of operation, we need to anesthesia, we need command. It's a special condition. We need to take care of our patient during a major stress condition. Regarding the gene therapy is still difficult. There are few over the world gene therapies for specific diseases. It's promising, but still, because mitochondria is expressed in all of the organs of the body with the different ways and different percentages. So you need to find this vector that it will go and spread everywhere to give the gene, to deliver the gene to the target cell. It's still difficult. It's still in the beginning, but is there some, let's say, some progress being made these days? We hope to hear the good news, inshallah, in the future.
Thank you so much. There's one question which says, with the next generation sequencing, one can sequence the mitochondria so deep that, you can reduce the heteroplasmy level. So is there still place for muscle biopsy, or would the testing in blood be sufficient?
Sufficient, definitely. If you are not sure about it, if the result does not match the clinical phenotype, you might go for further testing. But if it matches it, why to go for muscle biopsy? Muscle biopsy just nowadays, even though we do need a biopsy rather than open biopsies for some cases, but still, when you have a result that is inconclusive or it's negative and you're still suspecting mitochondrial disease, otherwise no need.
Okay, thank you. What is the coverage depth of mitochondrial genome, Alison, in a VGS, which is covered at 30x?
At least 1000x coverage of the mitochondrial genome in a 30x whole genome. Yeah.
Perfect. There is one scientific question. I think since we're running out of time, this would be the last one. Why are mitochondrial DNA more prone to mutations than nuclear DNA? Is there no repair machinery? Because what is the benefit of not having a robust repair machinery in place in terms of evolutionary context?
Okay.
Difficult.
Okay. Yeah, that's a great one. And it gets to the point of, as adults, we can be accumulating these mitochondrial variants, right? Because there is more error than the nuclear DNA. So there are fewer repair mechanisms in mitochondrial DNA. They're not folded around histones to protect them. And they're right next to all of those radical oxygen species being generated. So all of those things together mean they are going to be prone to more errors. There are some selections against mitochondrial errors, but yeah, they are. I think, why evolutionarily would they not? Because they are redundant, because the mitochondrial DNA is redundant in each cell, you can tolerate more error. That disease threshold is higher than a nuclear DNA variant. And there are some selection mechanisms in the replication process for getting rid of some of those large-scale deletions that are accumulated as an adult. Does that cover the entire question? That's a big question.
Yes, it was wonderful. So I think we should close the session now. I would like to thank the speakers. I was absolutely fascinated with the content and also with the case studies that we presented here. And I hope everyone shared the same feeling for the webinar. There's one of the questions where we asked as a poll whether you learned something new from the webinar that you didn't know before. That would be really important for us to just get a feedback on how valuable the participants found this webinar. So thank you very much. I think the handouts would be available for you to download. If you go to the bottom section, you will have a segment where you say share, or maybe in the apps, you can go to the handouts and you can download it directly from there. So thank you very much once again to the speakers and to all the participants. I look forward to the next session again. Thank you very much. Bye-bye.
Thank you. Bye-bye.